unfortunately formality. So Ronen received his PhD in physics and astronomy from Tel Aviv University, and he did a postdoc at Princeton. And from 2006, he's a PI at Ben-Gurion University. This is
למה התחלת לפני הזמן? תגיד לי, מה, אנחנו לא במזרח התיכון?
עכשיו קצת
מההתחלה. Professor Ronen Segev from Ben-Gurion University pioneered research of navigation and spatial representation in fish, and I'm sure he's going to tell us all about this in a few minutes. So let's
Okay, thank you. It's great to be here. The previous time that I gave a talk to ELSC, it actually was on the building just next to this building, and this place was a construction site. It was a huge construction site, and it was very fun actually to watch what was happening here back then. So I'm really glad to be here again. And I'm going to talk about mainly neurobiology of fishes, but I'll actually start, this won't work. I'll start with So, a few words about my lab. So, we are interested in neurobiology of fish, and one part of the lab is working on the archerfish, and the other part of the lab is working on navigation in goldfish, which is going to be the main topic of the talk today. I'll actually start just by introducing you the archerfish. Archerfish is a fish that lives in water. This is something which is very common. What they are famous for is their ability to shoot down insects that rest above water level, here. And the fish shoot at the insect a jet of water from the mouth, and they can actually grab them, bring them to the water level, and feed on them. So the cool thing about the archerfish is that you can actually train them to shoot on things that are artificial. Either they are presented on a computer monitor or some artificial object. So basically you have the fish equivalent of a human or a monkey who sits in front of a computer monitor and press buttons. And then everything that you can put into this multiple choice question, you can actually study with the archerfish from visual search, something that we worked on many years, color vision in fishes, and whether fish can count. They count 1, 2, 3, 4, and then they start to get confused. So one of the classical questions that raised by looking at the archerfish is the fact that when the fish look on an insect from below water level, then the insects are actually displaced from their true location. And the reason is that the water the light actually bends at the air-water interface, and actually the contribution of this refraction is actually something that the fish needs to take care of, because when they look on the insect, they are actually displaced from their true location. For example, here in this example, you have the actual target, and then according to the Snell's law that many of us learned about in high school, the information that is coming from the actual target is bent towards the perpendicular line of the air-water interface, and the visible target is actually displaced from the true location. And one of the classical things about the archerfish is that people used to give talks about this animal. So I used to give talk about the archerfish, and people used to come and approach me and ask me, okay, so how do they correct for the air-water interface change? And one of the things that you can actually think about is that they can correct by actually doing motor adaptation. Of course, there are two extreme ways of handling the issue of refraction of light at the interface. One is actually, if you think about it, somewhere in the genome of the archerfish, the Snell's law is encoded, and the fish actually use it as a, basically as a lookup table in order to decide where to shoot when it sees the target at some location. This is one possible explanation. This is the bird's refraction in the projector. I think it's not, it's there, it's okay.
Just open the door.
Okay. So another way actually to handle the issue of refraction is by motor adaptation. So, and basically, and this is, if you think it's, looks like the classical motor adaptation experiment where actually human subjects are endowed with goggles and they are presented with the ball, and they need actually to shoot the ball at the target that is on the wall. And initially, when there is no goggles, so there is no displacement, then they do some error after some training, and then they put on the goggles, and these goggles contains prism, which actually change the direction of view, and they continue shooting, and initially they make large error, and after some time they manage to adapt and actually converge into lower error over time. And then after some time, they actually can take off the goggles, and there is actually an after effect where the arrow goes in the opposite direction. Now, without the goggles, I'm adapted to the goggles deflection, and now I make an arrow in the opposite direction. And this is actually a classical motor adaptation experiment, and the question is whether we can actually test whether the archerfish use motor adaptation in order to compensate for the refraction of the light. And here actually comes the idea of the experiment that Sveta Volotsky did. She decided to actually present the archerfish with a target and add airflow above water level, and the airflow can be absent, and then the fish actually shoots at the target, and you can actually turn on the airflow and see what happens. Okay, and this is in this way we can actually recapitulate the classical experiment of motor adaptation done in humans, also in fish, and see whether they can actually use motor adaptation in order to correct for errors in the shot. And this is actually the way that it was done. We had a baseline where there is no airflow, then we have a perturbation where the airflow is present, and then we have a washout experiment where we remove the airflow and see what happens. And these are actually two examples of such an experiment. So the first attempt after the airflow is turned on, so airflow is coming from right to left. The target is here, so there is an iron net, and on the iron net there is something that looks like a coin, which is about half a centimeter disc, and the fish needs to shoot at the disc. The disc is tethered with a nylon wire, so it doesn't go off too much, and the fish actually see whether it eats the target or not. If it eats the target, then it gets a reward, and if it doesn't eat the target, then there is no reward. Okay, so the first attempt after we, so the archerfish can actually be trained to do this type of behavior. They shoot very accurate, and the first trial after the turning on the perturbation, the fish shoots, the perturbation comes from right to left, and the fish indeed makes an error to the left. Okay, let's see it again. Shot now, and arrow to the left, and then you let the fish continue trying, and after a few, this is on the 5th attempt, the fish is shooting, and you can see that the disc is deflecting. Okay, let's see it again, and you can see also the hand of Sveta rewarding the fish. Let's see it again. Shot, and there is deflection. Then we can actually do it shot where the airflow is coming from the back of the fish, and then we can actually do the baseline, the perturbation. You see that the arrow is going up, and then after some time it's going down, and washout, the effect is very small. You can see this is one fish, it's going in the opposite direction, and here is again baseline. Perturbation starts on and then dies off the error, and then if I do the washout, I see an error in the opposite direction. The same thing is shot against airflow. Here again you see a similar effect. Okay, so this is actually combining the data from all the fish just to see that the data is noisy. It's unlike the human very nice and very clean experiment. The fish shots is much more noisy, but still you can see actually the effect, and you can put do the analysis and convince yourself that they indeed initially after the perturbation starts they do a large error, then it dies off, and then you have in the washout you have an error in the opposite direction. Okay, so just to summarize this part of the talk, basically we've shown that there is a motor adaptation in the archerfish and are capable of motor adaptation. This is of course, in a sense it's expected, because if you shoot and over time even, well, the fish grows and the motor system changes, so you should have some mechanism of motor adaptation. Yes?
So you have, what you said is there are 20 trials a day. So the adaptation, is this on a single day? I mean, do you see savings from the next day?
Yeah, okay, so it's too noisy to see saving. So the adaptation is, you have a baseline perturbation baseline, and you can keep it between 20 to 40 fish for the champion fish. 20 to 40 trials for the champion fish. It's very hard actually to see saving, and we try to look for saving between different days. It's not there.
So all of this is, the adaptation occurs within like few trials?
Yes. Yeah, definitely. Yeah. It could be cool to see savings. Eventually what we found out is that these experiments are extremely hard. Okay, it took years to convince the fish to, you really need to fine-tune the amount of food. They are getting food every time they participate. Okay, this is a bottleneck. Okay, so archerfish, I haven't shown it, but you can actually enforce the archerfish to do adaptation in one direction and then do it in the opposite direction. Okay, and it seems that they do the adaptation in egocentric reference frame. And it could be a nice explanation, possible explanation for the light refraction at the interface. Now, keep in mind, it's a possible explanation. It's not, of course, a proof. And the reason is that wind, which actually generates the perturbation in our experiment, is also a natural phenomenon. So it might be that the archerfish has the capacity to do motor adaptation for wind. This is definitely a possibility. Yet still, this is really a natural system. Okay, the archerfish really, the adaptation that we see here is relevant to the real system in the wild. And this is unlike many of the adaptation experiments that we do in humans, where we move a manipulator or put on goggles and observe the effect. Okay, so this is one thing, and this is one part of the lab. Now I want actually to move to the main topic of the talk, which is navigation and representation of space in fish. And here we work mainly in goldfish. But while I'll actually show some ideas about where actually to go with this project later on. Okay, so navigation is definitely a very important behavior, because animal navigates all the time to find food, to find shelter, to escape from predators, and it involves aspect of memories, because many aspects of navigation rely on the fact that I learn something about the environment and I use this information later on in order to navigate. So there is actually a tight link between navigation and memory, and this is actually made navigation a gold mine from the point of view of brain science, where we know about place cells, grid cells, border cells, and direction cells that were found in the hippocampal formation of, initially it was in rats, and then of course in many other mammals, many other mammals. And basically, these cells constitute the system of representation of the animal position in the world. Okay, and there is a tight link between understanding the neural basis of navigation and addressing the issue of representation of space in the animal brain. Okay, so this is what we need to know before going to talk about fish navigation. And of course, we were interested in navigation, and the animal that we have decided to work with is goldfish. And the cool thing about goldfish is that you can actually purchase them from this size to this size, depending on the size of the requirements of the experiment, and they are very hardy. Okay, you can actually put them in all sorts of conditions, and they continue to live. It's on the level that you can, if you need actually to transform a goldfish from one place to another place, which is, I don't know, 2 hours away, you can actually just take it with no water and put it in the new place, and they will survive many times. So the first thing, of course, is to convince yourself that goldfish indeed live. You can actually do it in the classical way of letting them navigate in a plasmaze or something like that, and you can do it in a more exciting way, which is letting them actually drive a vehicle on land. And this is what we call fish-operated vehicle. Basically, this is, if you want, it's a car or a robot. The fish is housed in a water tank, and we have a computer and a camera that take video of the fish. And when the fish is approaching one of the water tank walls, then the computerized system drives the system of wheels on the bottom, so the vehicle will go in this direction. So if the fish wants to go in this direction, it will approach this wall, and then it goes in this direction. And if it wants to go in another direction, then the fish approach the wall, and the fish actually drives in this direction. And this is how it goes. Okay, so the fish is approaching the wall, and you can actually let it drive the vehicle. You can actually take it out. And the first thing, of course, is to be convinced that indeed the goldfish control the vehicle. And in order to do that, they actually need to overcome 2 issues. One is the issue of motor control. By driving a vehicle, and if you learn to ride a bike or drive a car, you know that there is actually a difference between what I do in the motor aspect of the driving and what actually the consequence. Okay, so the fish needs to overcome this. And the other thing is that they need to navigate in the new environment. Okay, and for them, basically, this vehicle is like a submarine for us. Okay, and in order to convince that they can do actually both tasks, is we do this type of experiment where they start in the middle of the room, and they actually trained. If they drive to this pink polygon, then they get a piece of food. Okay, and you can see actually the trajectory, and you can actually start after. They work in sessions of 30 minutes, eat as much as you can, and so you let them do that, and after something like between 10 to 15 sessions, they actually master the task and can actually drive to the target. And from time to time, you can see something like that, where the fish drive, and then it gets stuck, and then it doesn't get any reward, and then it actually managed to correct. You really see that they master the task of driving the vehicle. And of course, you can actually measure it by, you look on fish success improve over time. This is the first day. This is the starting point is in red, the green is the last point. Keep in mind is that when you drive in an enclosed environment, even a random walk, even a silly strategy will get you reward. Okay, so this is something to keep in mind. And initially they get almost no reward, and this fish actually get no reward in the first day, and then they actually start to get more and more pieces of food over the session. And you can actually, if they are indeed driving, over time they get improved in driving the vehicle, because if you start at the center, you get go straight to the target, then you get the reward. Yes.
Excuse me, I hope it's not a nasty question, but
yeah.
What is the advantage of going through the vehicle and not doing it through?
Yeah, it's not a nasty question. This is actually just a proof of concept. The main idea, and it still does, is actually the fact that you can take the fish and let it navigate in large environments. So my lab is confined, and you can actually take them out and let them see how they manage in order to overcome the issue of navigation in a very large environment, like going out here. Okay, so this was actually the first motivation to do it. It's still the motivation. We still work on it. You can actually do other things like motor adaptation with this thing that we have done. Okay, but the initial thing was actually let's break the walls of the lab. In the lab we have a water tank which is 1.4 by 1.4 meters. this is the largest water I can keep in the lab. Okay, this is the motivation. Okay, so this is just starting from multiple locations. They get to the right place. And the question is of what have we learned from this fish-operated vehicle. The first thing is that we found out that fish became proficient in driving the fish-operated vehicle. They can navigate in alien environment, and this actually also tell us that goldfish can navigate, and you can actually show it in other less high-tech way of navigation. I haven't shown you the data, but they don't use a single visual cue in order to navigate, but they use actually a combination of cues, and they can navigate from multiple starting points and adapt to manipulation of the cues. The next step is, of course, after we establish the fact that goldfish can navigate, we can actually ask what is actually the neural basis of this navigation, and the core of actually asking this question is actually to look for representation of the position of the animal in the brain. Okay, so it's basically looking for the representation of space in the brain. And here actually I would like actually to take a step back and learn something from the history of places. And the first lesson that we need is actually we need to address the issue of representation of spatial information. This is one thing. The second thing, and this is actually might be more important, we need information about where to look for this representation. And actually there is a very interesting story by James Rank back in the beginning of 70s, 1970, 1971, was doing the exact experiments that Ostrovsky and O'Keefe were doing by placing electrodes in the hippocampus of rats and looking what happens. And you read one of their paper, you read it, and the description is of a placer. And the issue is they missed actually the fact that they were looking at placers because they were not aware of the connection between actually the hippocampus and memory, and moreover, hippocampus and spatial memory in the rat. So this is something that O'Keefe and Ostrovsky were aware of, and this is why they actually understood we are looking at the representation of location by measuring activity in the hippocampus. And this is actually something which is very important for us because now we take this goldfish. Goldfish was evolutionary diverged from us mammals 450 million years ago. So this is a totally different design of a brain. Okay, and the question is where to look for the representation of space. And for this, we actually have 3 lines of evidence. The first one is development. The second one is molecular marker and transcriptome. And the third one is functional neuroanatomy. I should warn you that I'm going to tell you now a story which is going to be confusing. It's not going to be a very clear-cut bottom line. The task is clear. We want actually to find the evolutionary and developmentally counterpart of the hippocampal formation in mammals. Okay? The answer is that functional neuroanatomy is really, or comparative neuroanatomy is not a crystal clear subject. So I'll describe the data, but the bottom line is going to be somewhat confusing. Okay? So the first lesson is actually coming from development. The development of the fish brain, and when I say fish, I meant teleos fish. Teleos fish is about 90% of fish species, and they about 50% of the vertebrate species on Earth. Okay, and the development of the teleos brain starts like our brain from the neural tube. So you have the neural tube, and in our case, what happens is the neural tube is actually folding, and the regions that ends up inside, we call them hippocampal formation. In teleos fish, the neural tube starts the same, but then, I cannot do it with my hands, the folding is not inside, the folding is outside. So basically you have evagination process in us and eversion process in teleos fish. So basically the regions that are on the side of the brain, the side of the pallium, correspond from the point of view of development and evolution to the regions that are inside the brain. So it's very tempting to go here and say, oh, I found the hippocampus of the goldfish. Okay, so, and indeed, there is actually one group from Spain that did a classical plus maze experiment. They let goldfish to navigate, and they lesion the side of the brain, and if they lesion the side of the brain, the goldfish fail to navigate. If they lesion other parts of the brain, the goldfish managed to navigate the plus maze. That's very nice. We found the hippocampus of goldfish. The issue is that you look around, there is another group from Japan, and they do a similar experiment. I use this map. If they lesion the side of the brain, goldfish navigate nicely. If they lesion the top of the brain, the goldfish fail to navigate. It's a similar task. It's not exactly the same task, but definitely it's a navigation task. This was actually so important because these two results definitely contradict. Okay, if you lesion the side or the top, one group claims it's the side, one group claims the top. So I asked Shachar Givon in my lab to do again the experiments. This is the learning phase, and then you can do the side lesion or the top lesion and the control, and you see that actually if you lesion both the side or the top, then the goldfish actually fail to navigate. So it seems that both groups are correct. Okay, so now we left with the issue of, okay, so it seems that there is no clear functional neuroanatomy lesion experiment that I can do that will state this is the hippocampus of the goldfish. Another way of obtaining information, this is actually the third attempt in our lab, is to do spatial transcriptomics. So spatial transcriptomics, basically what you can do is you can take the brain, you can dissociate it into cells, measure messenger RNA level of expression in each cell, and then actually find cell types based on their single RNA sequences. And you can actually do it also in slices. Okay, so if you do in slices, you can actually do what is called spatial transcriptomics. Basically, you at each small region in space, which is about 50 by 50 micrometers, you go and do the same transcriptomics analysis, measure RNA molecules, and then you can actually try to divide the different regions in space into different regions in the fish body. And if you do that, I'll skip. You can get cell types for the cell type atlas for the goldfish brain, and then you can do the division. And then at the end of the day, you have this spatial transcriptomics data, and this is cross sections of goldfish and telencephalon, the region in space that we are interested in. And you can actually look for expression of single molecular markers. And one of them can be actually GLUT1 and GLUT2. And what you find out is that GLUT1 is expressed on the side of the brain, which corresponds to the region in space where the Spanish group claimed this is the hippocampus of the goldfish. And why this is important? Because this same marker actually expressed also in part of the mammalian hippocampal formation, which is the subiculum. And in the subiculum, actually, it was found that there are cells that represent aspect of space. So this actually looks like this is this fits nicely, and but of course this is with a grain of salt. This is based on few markers. Here it's one marker, and there are clones of genes in the goldfish brain. Goldfish actually went through two old genome multiplications, so they have about 80,000 genes in their genome. Okay, so really take it with a grain of salt. So going back here, as I said, comparative neuroanatomy is not a crystal clear subject, so there is no clear hippocampal formation homologue in the teleos brain, in the goldfish, and of course in the teleos. Still, we can actually even if there is no clear, we can actually, there is something that we know, is that if you look on the polyum, if you lesion parts of the polyum, then the capacity of the goldfish to navigate actually drops. Okay, so we can still follow the O'Keefe and Ostrovsky recipe of going into this region and look for the representation of space. Okay, so in order to do that, of course, you need the capacity of recording from a fish while it's swimming. So we developed this thing, and this is work by Ludwig Pinsky that developed this implant. The implant contains a neural logger, which is about 2 grams in weight, and a battery, of course. And there are 4 tetrads that go into the brain and allow us to record single cells in the goldfish polyum in regions that we think are associated with navigation. And in order, we do this experiment while we record actually the position of the fish by a video camera looking from above. This is how the experiment looks like. This is a 5x speed of the goldfish are swimming slow. They don't super excited like this. And then what we can actually do later on is correlate the activity of single cells that we get and the position of the animal. And we found out actually several cell types in the goldfish polyum. One is H cells. These are cells that are active when the animal is near the border. Blue is low activity, and reddish is high activity. There are eight-direction cells, like in mammals, cells that are active when the animal is looking in one direction and less active when the animal is looking in another direction. There are speed cells, cells that actually correlate with the speed of the animal. Speed is the absolute value of velocity, and there are actually velocity cells. This is the velocity space. This is velocity along x direction, and this is velocity along y direction, and there is a bluish color in all directions except in some region in the velocity space. So basically this is a cell that is tuned both to the absolute value of the speed and the direction of speed. If you want, it's a conjunction of speed and direction representation. Okay, so this is one type of cells. Another type of cells that we found are boundary vector cells that look like this. What you will hear is actually the activity of cell when the fish is swimming. Okay, so this is a different experiment. So basically we are looking from the side. The water tank is 70 by 70 centimeters in size, and it's about 20 centimeters in width. Okay, so basically the fish is more or less confined to move in a two-dimensional world, and then we can actually plot the trajectory of the fish in black. And every time we detect an action potential for the particular cell, we draw a red dot, and you can see that there is a cluster of red dots on the left, and there are almost no red dots on the right. And you can see also the heat map here, red colors on the left, blue colors on the right. Basically, this is what is called in the mammalian world boundary vector cells, cells that actually represent space. But actually it's next to the wall, and this is another example of boundary vector cell. Again, you look here the spikes. Here spikes occur when the animal is on the top, and do not occur when the animal is on the bottom. And you can see it actually both in the scatter plot and in the heat map. Okay, so this is very nice. So this is really show us that indeed there is representation of space in the goldfish polyum. One thing that really, when you look on this type of activity, really that comes to your mind, and maybe this is a representation of the fact that fish actually, when they dive, they express, they actually experience larger hydrostatic pressure. Okay, so you should expect that fish has some capacity to measure the hydrostatic pressure. And the cool thing about hydrostatic pressure is that it gives you the z coordinate, the vertical coordinate just by sensing it. Okay, you don't need to learn something like that. You can sense it, and you know where you are in the water column. And it's very tempting to ask whether this type of cells that we see here, they actually tune in some sense to pressure. Then actually there was work from the lab of Teresa de Pereira in 2021, where they show actually that in the behavioral study that fish indeed can measure absolute depth using hydrostatic pressure cues only. Okay, so they just remove all other cues. The only thing that was available for the fish was actually hydrostatic pressure, and they can do it. And they even discuss about possible pathways of how they can do it. One of the things to know about Tilos fish is that they have a balloon inside the belly that is used for adjusting the density of the fish, so it's going to be neutral. So they don't work against gravitation when they go up and down. Okay, and so basically this gas chamber can actually be used in order to sense differences in the hydrostatic pressure, and this is something that the fish can use by integrating it in actually to find the absolute position in the water column. Okay, so how can we test that? So we spent some time to build this gadget where we have actually a pressure chamber where you put the fish inside, and there is a system of electronics and a compressor that can change the pressure inside the pressure chamber. So basically the fish is swimming, and we change the pressure, and the fish actually feel it. And you can actually then try to look for cells that are tuned to pressure. And this is there are two examples. This is pressure, and the pressure was selected to be changed identically to the 70 centimeters water tank that we used before. And this is the Fahrenheit, and it can go up and down if you take the pressure up or take the pressure down. You can also see things like this. So this is another pressure-tuned cell, but when the pressure is going down, there is one tuning, and when pressure is going up, there is another tuning. Okay, so it seems that there might be hysteresis in this type of representation. The next step is, of course, to ask, okay, so how does it actually translate into the actual swimming fish? Okay, and in order to do that, we recapitulate the experiments by Teresa de Pereira, where we let the fish swim in the same water tank that we used before, but now we put a ceiling on top. The ceiling is transparent from the point of view of water, but the fish doesn't see it. Okay, so you can actually pour water from above and change the pressure in the environment. So the fish is looking around, it sees the same cues all the time, but pressure is changing from environment 1 to environment 2. Okay, and then you can actually measure the activity of single cells, and you can see that there is tuning. The tuning is similar to what we have seen before, but it's modulated when I change the pressure in the environment. So the bottom line, we're still in the middle of the project, but it seems that these cells are not pressure-tuned cells. Okay, they are modulated by the pressure. They represent space, and if pressure is changing, then they change their tuning, but not in a dramatic way. Okay, so it's not a pressure-tuned cell. The pressure is a cell that really tuned only to pressure, and this is not the case here. Okay.
So this is the actual Fahrenheit, or is this normalized?
Yeah, this is the Fahrenheit in goldfish is very low. It can go up to a few hertz at most.
So how fast do they go? I mean, is it informative? You know, if
yes. So one thing that it can be informative is that if you have 10 cells like this, then you are okay in terms of signal to noise ratio. And the other thing is goldfish, they swim very slow, so they don't do some dramatic thing. These animals were domesticated in China about 2,000 years ago. Okay, so these are not wild animals. Okay, okay, so this is and really the last part, and this is This is into the future. This is the future outlet. Where do we go from here? The cool thing about fish is that you can actually imagine going to the natural habitat of the animal and do electrophysiology. This is something that you actually cannot do in other animals. Okay, it was done in natural environments in the past, but not in the natural habitat of the animal. And the reason is that you can actually, if you look on the issue, the main issue is, at the end of the day, is the size of the battery. Size of the battery really limits the capacity of doing long experiments. And in fish, you can always counterbalance the weight of the battery with a float. So the fish will not feel any weight, it will feel drag. So if I try to swim fast, then it interferes, but I don't feel that there is something above me too much. And this is really this brings the fact that water can actually allow week-long recording sessions. Just add a float in order to do a long experiment. And you can actually get information about the position of the fish by using acoustic tracking. Basically, you put an acoustic pinger on the fish, it pings, and there is a set of hydrophones that pick up the ping and tell you where the fish is located. And you can add a motion sensor that really provides the behavioral information, if the fish is snapping something or doing something, and also the head direction information. And this is actually, all this can be done in this animal. This is the northern pike. It's the top predator in many lakes in North America and Europe. And the lake that we decided to work in is Ormstrup Lake in Denmark. This is how it looks. It's on a good day, it looks like this. Most of the time, it's gray. And the cool thing about this lake is that it's equipped with a set of receivers that allow you to detect the position of the fish. Basically, you can do the experiment like this. You catch a fish in the lake, and then you implant a logger and the battery on the fish head. This is how it looks after the surgery. You have it's about this size fish, and it has an implant. Then you release the fish to the lake for 4 days, and here this is this was really the peak of the experiment, where we had 5 fish, and you can see the last half hour of the trajectory of the 5 fish in the lake. And at the end of the 4 or 5 days, you go and catch the fish, because you know where it is online. So you go and actually grab the fish from the water tank, from the lake. It's not that easy as it sounds. And then you can actually correlate the activity that you measured from the brain and the position and the behavioral data. I was very tempted to put here actually data, because in the last week I analyzed this data, and then I looked, and then I said, oh, this looks exciting, but then I said, no, it might be that I'm wrong, and I need to I need to rethink. So this is the next time that I'm going to give a talk here. I think this is going to be the main topic of this talk. Okay, so let me conclude. We showed that goldfish can learn both to operate a vehicle and navigate on land. The mapping between teleosteal cephalon structure and other vertebrates' brain is an open issue, and I must admit that I don't think it's going to be settled. So if you hear a talk, and you're going to hear a talk in some of the conferences, we work on the hippocampus of the fish. I don't think this is right. We found indication for encoding of space in the goldfish brain, and it seems that hydrostatic pressure modulates firing rate of cells in the goldfish and encephalon. Of course, more evidence is needed here to establish the existence of pressure cells or pressure modulated cells. And another thing in the issue of pressure cells is that find a neural pathway that allow information from the sensor, whatever it is, get into the telencephalon, where it can be used for navigation. Okay, so the people behind this work are Ehud Vainpinsky, Leo Cohen, Chachar Givon, Tal Samina, and Svetlana Polotsky are the main figures around, behind most of what I've shown. And Stal Bitton, Muhammad Tibi, Anna Ochberger, all in collaboration with Amit Seisel Lab. This is the transcriptomic project, the funding, and thank you for listening.
Do fish also have this drift in the representation? Like I know that place cells, for example, in Ronen, sometimes they change, the population of cells that act as place cells change, and when they return acting as place cells, they tend to encode the same place in the environment. Does it happen also in fish?
Yeah, the question is whether there is remapping. It seems that there is remapping, although the evidence is not very strong as in the rat. One issue when working in the goldfish to keep in mind is that it's a less stable system than the rat. And the reason is that the skull is much thinner. Okay, so thinner skull results in more motion. Fish also, goldfish, the brain doesn't fill up the endocast, so there are more motion artifacts in this type of experiment. The top that we managed to do was this type of experiment with the pressure, is where you have environment 1, 2, and then environment 1. It's really it's a very hard experiment. Okay. Now that we move to the northern pipe, all of a sudden you see a skull which is about 4 times as thick as the goldfish, and all of a sudden you say, oh, it's much more stable. So there is the issue of stability, at least the goldfish.
What's your conclusion or your intuition about the evolutionary aspect? So it's a convergent evolution story?
I think it's probably convergent evolution story, because if you look on the stem of the phylogenetic tree, over there are not too many animals that navigate in a way that requires memory. Okay, so it's good. You ask me to speculate, I can say whatever I want. My guess is that it's convergent evolution. And this actually, it also, there are actually very nice results in zebrafish larvae from the group of Drew Robson and Jennifer Lee. They found places in the larva. And over there, it's really all the pallium is filled up with cells that are, sorry. So over there is no localization of this representation, okay, in the brain itself. Unlike if you look in the in the rat, there is a localization of function. Okay, so it seems different system. This is my guess.
What about the local field potentials? Can you record them? And can you see something related to classical hippocampal signature?
Yes, so I haven't showed it, but in the case of the boundary vector cells, you can see actually that there is linkage to beta rhythm. So it's not theta, it's beta, but definitely it exists. Okay, so you can see beta rhythm and activity, and which is, I think it's interesting. I must admit that I'm not very fond of the whole beta story in the rat. Luckily, I don't work with rats, so I can just look and be critical. But I think it's an interesting observation.
This area with the cells that you showed, is it close to sensory areas, motor areas?
No,
How is
the
it
telencephalon
related
doesn't get direct input of sensory information except from the olfactory system. In the experiments, we usually try to evaporate the olfaction cues by mixing the water. Okay, part of it because I don't know how to control it. So it's very important to mix the water, and then you're okay.
And water parts is close to the water?
No, it's, there are actually there are stations outside of the telencephalon that project to the paleo, but there is no direct link. At least, well, keep in mind these are fish. The neuroanatomy is much less. No, okay, but and the diversity of fish is too large. So it's not that okay, I look on the zebrafish and zebrafish is convert. It's a miniaturized version of the goldfish. They're not. If you look, goldfish brain is much more complicated, much more complex than the zebrafish. Although they are not very far away in evolutionary terms. Okay, they are actually sitting on the same branch of the Tibios evolution.
Regarding this extremely low spiking rate, is this something unique to this species, or maybe it's temperature dependent?
One thing is that in the optic tectum we see higher. Optic tectum is really, it's called optic tectum, but it's a major sensory processing unit in the fish brain. Okay, so it receives actually information from other senses too. So over there you see a higher rate. So I think it doesn't mean it's not temperature probably, because it's the same temperature. It might be that something that I don't think the goldfish is the place to show it. Goldfish telencephalon is small. It might be that you have spiking neurons, but you have also other types of information processing, maybe a graded potential. I don't know. This is something that electrophysiology of the type that we do is not the way to show it. This is a speculation that I have in mind for some time. Okay, so I don't think it's temperature. I think it's the region in the brain.
Okay, so thank you very much.
לעכב אותו שהוא לא יוכל להגיש את הקלאס?
לא, לא, לא.
לא בסדר.
אבל המזל הוא שכן אפשר לעשות מיפוי סביר במה שאנחנו מדברים. הפחד שלי כשנכנסתי בספר של שרי מזמור, יש שם גדול פלייסנס. היא כתבה ספר ב-2006, פלייסנס. דקה לפני שהמוזיקה I can speak to English. So Shani Mizumuri wrote a book in Moses are going out of Britzels about places. The book is obsolete almost immediately, and the preface was actually written by Rank, and the quotes from a paper they wrote back in the beginning of the 70s. And you read the description, they look on places that just don't know that. And I was terrified that we don't know the neuroanatomy, anything, and this is not good. The good thing is that the Spanish group actually did a very systematic work. They started by removing the telencephalon and then goldfish. Don't navigate at all. And then they start to dissect. Okay? And it seems that there are some regions in space, in the brain, that are associated with navigation. And this was actually critical because then you know where to place the electrode. There is now another work from zebrafish where they actually look on the front copy from the tail, which is projecting somewhere in the midbrain, and this actually represents what the fish thinks it's going to be based on what it's tried to do with the tail.
Yeah, giraffe has some.
It's very similar. She's both.
Yeah, so this is a different region. It's not the telencephalon. So this is actually how the story went.
Thank you.
Okay, bye-bye.
Really awesome stuff. I mean, the first part of your talk was, that was really, that was very new to me, the artifish stuff. I wanted to ask about, so the pressure sensor part actually confused me a little bit.
Yeah.
So, like, how should we interpret that? So, do you think that some of these border and boundary coding that you're seeing, can this be a confound of pressure sensing? Because the deeper you go, of course, the pressure would increase. Would that be like a surrogate readout? Like, is pressure going to be confounded with boundary vector?
I think the confound word, maybe it's too, it's going to be conjunction with.
Or you think that there is really a pressure part to it and a spatial part to it? Like,
No,
or
I think there are actually.
and both are like in a conjunctive manner.
Yeah, they are conjunctive manner. This is why I think it's better to call these cells pressure modulated cells, because they have their tuning, okay, which is more or less boundary vector cells. It might be there are actually places, I don't know. Just it might be that the water tank is not big enough.
Right.
So definitely there is a place tuning, but this place tuning is tuned by pressure. Which makes sense if you are fish. Okay, it's not. It doesn't make sense if you live in air, because in air the pressure gradient
doesn't change
nothing.
much.
It doesn't change much. You really need to dive a few meters a second in order to feel it. Okay, this is why when you go into a very tall building, when you take the very fast elevator, you feel it. Okay, otherwise you have a problem actually perceiving the pressure changes. Okay, so I think that the right term is pressure modulated cells. It's not pressure tuned cells.
I see.
Okay.
And another small question I have about the artifish is that, I mean, of course you can negate the refraction if you're standing exactly below the target. So when they shoot, is there a tendency to go close to the right angle?
I haven't observed that. If you look in old papers, then there is the claim that they counter it by going into the just below the target. I don't see it.
You haven't seen it?
Yeah, it's it definitely exists in old papers.
I see.
It's not that there are too many labs working on these animals.
Which is a good thing for you.
Good and bad. It's good because you don't feel the pressure like in the rat. It's bad because you don't have the the feedback is good. Feedback is good because you read it and you give me feedback. It's good. There is no synergy because one lab can do neuroanatomy and one lab can do electrophysiology and one lab can do behavior. We need to do anatomy, behavior, and neuroanatomy. This is the drawback of working on such cool but not very popular. Yeah.
Okay, awesome. Thank you so much.
Bye bye.
Bye.
Hey, how are you?
How are you?
Hi.
Could you explain again the difference between the two different types of velocity cells? Goldfish.
You mean speed cell and velocity?
Yeah.
Yeah, speed is I care about that I travel 50 kilometers per hour, but I don't care if I drive to the south or to the north. Okay, this is speed. Velocity is speed in the physical. When physicists will talk about speed, they talk about velocity. It's a vector that cares about both about how fast you go and also in the direction. Okay?
And so those two things are encoded
In two different cells. Yeah, there are cells that look on the absolute value of the velocity, which is the size of the vector, and this is the speed. And there are cells that care about the direction. The funny thing is that when I told it to Edvard Moser, he said, yeah, but he told me something like, yeah, just it's better that you know that speed signal you see in many places in the brain. It was it was very it was unamused by the speed cells, and he is probably correct. But velocity is something that really it's more abstract because I really need to know that I go in this direction. Okay? Okay.
אתה יודע, הסיפור הזה של ג'יימס האנק, שהוא לא קלט שהוא מסתכל על... הוא כתב על זה בערך. הוא כתב ש... לא ספר, הוא כתב על זה שהוא... עשו לו טקס... לא משנה, כשהוא כתב על זה, אז הוא אמר שהטענה שלו זה שהוא לא היה מוכן.
נכון. We should have a prepared mind, we didn't have a prepared mind. כן.
אני חושב שאתה צריך גם כן לקחת בחשבון. כי יכול להיות מצב הפוך. מה זאת אומרת מצב הפוך? אתה מחפש לייסר, סייטרגל, תשמע, לדעתי, יש שם מערכת, יותר סביר להניח שיש שם מערכת אחרת. היא לא פחות מעניינת, אפילו יותר מעניינת, כן? וכאילו...
כן, אתה רואה שזה לא...
צורך חזק לא לאנוס, אבל לחפש את ה... לא, אתה לא מרגיש ככה.
לא, תראה, אני מדווח מה שיש לי. אצלי אין פלייסס. יש לי boundary vectors, שזה סוג של פלייסל, אבל לא מצאתי פלייסס קלאסיים. חיפשתי הרבה. חבר'ה במקס פלנק מצאו...
לא, כן, הסיפור הזה לדעתי משנה את כל הסיפור. נראה לי שיש פה עניין של ספישיז.
זהו, אז יכול להיות שזה ספישיז, יכול להיות שזה גם עניין של שיטה. הם עשו את זה עם רובוט מיקרוסקופי שרודף אחרי הדג.
כן, אז רציתי להגיד לך, הכניסה שלך של ה-self-navigating fish, פשוט תקליט מהפיש.
כן, זה נכון, זה דבר הרבה לעשות.
זה הרבה יותר טוב מרובוט שרודף אחרי ה... למה לא עשית את זה? זמן?
זמן, רק כך וכך דברים לעשות.
תגיד, אבל גם טכנית, כאילו, אם תשים מכסה, רואים שם
זה לא אכפת לדעת, הוא לא מקבל, אתה שואל אם הוא קיבל מחלת ים, התשובה היא לא.
לא, לא מחלת ים, זה משנה את התנועה.
כן, אז באיזשהו שלב שמנו מכסה, השילושין לא היה מאושר מזה והוא סלק לי אותו. זה בנוי עם מכסה.
אז סטודנט, פוסט-דוקטורנט?
לא, סטודנט, אז הוא היה מאסטר, עכשיו הוא דוקטורנט. באיזשהו שלב הוא מהנדס מכונות, הוא אמר לי, לא, בלי זה. שושין אמר לי,
אתה נפגש עם עוד מישהו?
כן, אני הולך עכשיו, יש לי אישה בינינו חברה מאוד טובה, אז אני יודע להגיע לשם.
בפעם הבאה שאתה רואה פה, אתה לא מכיר אותי מהמילה,
האמת היא שיש לי עכשיו לדבר שעה 20 דקות.
עכשיו לדבר.
Okay.
Lot of traffic?
Yes, here in Jerusalem it's too much traffic. But outside it's okay.
Okay.
Let's see. I was thinking like you're gonna say that through running.
I did.
You ran
I
and
did.
then you went to the party?
Yeah. I forced myself to get up and run this morning and broke it.
I'm skeptical that the
PowerPoint today?
yes, for the last class. Are you ready?
Shouldn't it be pretty short? 5 minutes.
That's 10 minutes.
No, 5 minutes.
It's 5 minutes?
It's 5 minutes.
12. I know how we're going to manage this in 5 minutes.
No, because they asked us to make 4 slides.
Yes.
So we kept it like exactly 4.
You did more?
I have 12.
No, it's
Did I
good.
make-
You say on the movement that you don't need a lot in the slides.
Yeah.
That's
You said-
interesting.
It says you should